Hello, everybody. I would like to thank the RTMFM for this opportunity to share and discuss all together the recent papers at the end of July, where 13 experts speak about new challenges in imaging cell biology. The question to all the experts was, what are you most excited about when it comes to microscopy in cell biology, and what challenges need to be overcome to reach these goals? And what I've done to prepare this talk, quite dense, I have to say, it's a search for each expert backgrounds and then a summary of their interview to produce a sort of identity card for each one that I will show you. And then I will end with some points of convergence to open the discussion, like a round table on the chat. The first I will present you is Emma Lundberg. She is director of the Cell Atlas and Human Protein Atlas program. And she is quite exciting about the convergence of approaches on genomic single-cell technology and microscopy. And this for her allowed deeper understanding of the relationships between genotype and phenotype. But then some obstacles be removed, like the understanding of the complexity of subcellular proteome, then also determine a multiscale proteome architecture of cell and its dynamic rewiring, and ensure the representation of cells in an unbiased way. This is really important. And the future challenge to reach this goal is have a strengthening community engagement with open science initiative and develop a self-driving microscope that are capable of long-term assay planning with optimal decision-making using deep algorithms, for example, and to predict some cell fate. Musa Malanga, he is also implicated in the Human Cell Atlas project that all of you know, and for him, the title is Spatial Single Cell Biology Revolution, and what he has to be improved for him is that we need a high throughput acquisition of spatial information that are not enough developed actually, and computational framework for quantitative analysis of high dimensional imaging dataset. And the future challenges is to have the spatial transcriptomics used as a routine in pathology and histology, improving in this way the diagnostic of biopsies. What about Brenda Andrews, professor at the University of Toronto, is that the high throughput plus the genome editing and plus the machine learning today can describe quantitatively single-cell biology. And she's using, for example, budding yeast and some genome and the genome of yeast and scale mutant as a roadmap model. And for her to continue, it's important to continue the production of an image dataset that sample cell biology in different environmental conditions and model system, develop deep learning, and also have what the biology community will need are solutions for storage of terabytes of imaging data, and also benchmark for quality standards. And for Jae Byung Chung, from professor in South Korea, he is specialized on expansion microscopy, and for him we have to go under 10 nanometer of lateral spatial resolution. We need the availability of small molecular levels of very few nanometers, and the obstacle to be removed is also the ultra multiplex of the imaging technique that have to recognize more than 10,000 distinct proteins. And for him, the expansion microscopy can solve all these issues. But the challenge is to expand the repertoire of targets recognized by small antibodies, there are few at the moment, to achieve ultramultiplexing imaging on 3D with a precision of very few nanometers, improve the registration technique with a precision of 1 or 2 nanometers, to manage dip distortion and due to the cycle of labeling and expansion, and introduce a barcoding principle to accomplish ultramultiplexed imaging of protein from the expanded specimens. And Lucy Collison, she is the head of electron microscopy at the Francis Crick Institute in London, and she is expert on imaging cells and organelles in their native context by using volume electron microscopy that can reveal nanoscale architecture of cell and tissue in 3Ds. Main problem is that actually we have the automatization of image acquisition, but this produces vast quantities of complex image data and hundreds of gigabytes data per day. So today there is the lack of availability of a large-scale training data, and weeks and months are needed to generate segmentation for a single organelle. And the future challenge is the contribution that the electron microscopy will give in clinical research at the organelle and membrane nanoscale levels, develop the correlative and multimodal imaging pipelines, and growing up a community of experts into these fields. That is also important. And what about Julia Mahavit at the MBL? She specialized also on electron microscopy and correlative multimodal microscopy that for her must be developed. And today the atomic resolution can be achieved by cryo-electron tomography that give us structural cell biology, and then fluorescence of light samples can be studied with the fluorescence imaging at the single molecule level in a space and time. But to correlate these two approaches, which is important, is to go further, have a better link cell and structural biology, and to realize data with unsurpassed signal-to-noise ratio. And to do this, she proposed to introduce a direct electron detection camera, automated high-throughput data acquisition, and the development of correlative light and electron microscopy in 3D. And what about Akihiko Nakano? He's the team leader of the live cell super resolution imaging research at the Center of Advanced Photonics in Tokyo. In his team, they are developing a new microscope. Is a super resolution confocal live imaging microscopy to have a high 4D resolution microscopy and follow the vesicles network in the live cells. But still we have some problem to be solved, like the ability to see also the unlabeled molecules, for example, using correlative light and electron microscopy. And actually, the problem is also that CLEM is quite laborious. So he suggests that it could be interesting to increase the use of correlative light and electron microscopy, improve the artificial intelligence processing imaging coming from CLEM experiments, and develop, for example, innovative microscopy without labeling at high resolution using other kind of light source, like soft X-ray. And also Dong Li, he has a question, a dilemma to study subcellular dynamics, is how to acquire spatial temporal information with high fidelity and quantifiability with less photobleaching and phototoxicity. And the obstacle to be removed is that actually gain in spatial resolution comes with trade-off in other imaging metrics like standpoints, lower frame rates, and smaller field of view. And he suggests, for example, to develop a super-resolution microscopy by implementing adaptive optics. This is just ongoing also. And it's important to have a holistic design that synergizes the development in both optical and also algorithmic and methodology, and synergize also hardware implementation with deep learning methods. Suyana Manley is head of the Laboratory of Experimental Biophysics in Lausanne, and she's focused on the dynamics into noisy structure-function relationships using super-resolution approach. And we have, for her, we have also the duality of the structure-function relationship that is boosted by cryo-electron microscopy, and the structure and dynamics, and in a chemical also physical context using biosensor, for example, that can be measured by fluorescent imaging. What is important is to be able to capture and functionally enrich and obtain dataset to decipher dynamic structural signature among the noise. And today there is the problem of the toxicity of super-resolution microscopy and the noisy of adaptive microscopy, what is due to the reduced light dose. And to go further is, for her, is the adaptive optics offer a first solution for the toxicity, and the deep learning allow then the reconstruction and the denoising restore with low light images. Johannes Schoenberg in San Diego is the head of the 4D cell biology lab. And for him, it's important, like he said, to increase the access to use of the lattice light sheet microscopy, who is provide both the low phototoxicity and the speed to capture high frame rate of 3D movie cells. And in his lab, they are applying these approaches to study the mitochondrial dynamics. And the obstacle is that today few quantitative analysis tools are available for the hundreds of terabytes that are coming from one single experiment. And it is important to develop pre-processing analysis, annotation, visualization, and long-term storage, and also increase the limited accessibility today to these techniques, to the adaptive lattice light sheet, and also have data analysis tools that coming together as a community. We are and what about Ting Wu, who is working at Harvard Medical School, and she is developing oligostome to study the relationship between genome organization and gene regulation. And for her, there is the convergence of a challenge, conceptual and technical one. For the conceptual one, we need to put aside a preconceived notion of what the genome looks like. And technically, we need to increase the optical and genomic resolution. What it is also important for her is, for the future, is to democratize technologies and reducing cost, rendering microscopy and imaging and image processing more accessible. And I agree with her. For David van Wallen, it is important to combine machine learning with imaging. And for example, today live cell reporters plus multiplexing are increasing the amount of information about the cells. And the machine learning increased the amount of information extracted from imaging data. So what is still a problem is that machine learning requires a large amount of labeled imaging data. And he suggests that we may have to rethink what it means to label an image. We need to integrate this new method into experimental design. And he asked if we can design better, more scalable experiments. So this will be a question that we can discuss at the end. And finally, Asaf Zaritsky, who is a principal investigator in the Lab of Computational Cell Dynamics in Israel. And I like this picture coming from his website, that there is, we need to have communication and interaction between the biological experiment for the interpretation of data or the computational system, and then have a method to discuss together this results. And for now, the deep learning, it's a powerful technique, right? And but is quite uninterpretable. So for him, it is important to develop user-friendly tools for efficient visual quantitative exploration that are accessible to the cell biologist and user as we are. I will conclude, just give you some convergent points that I identified of all these experts interview. And first of all, the improvement of 4D resolution, and we need to develop correlation between structural and functional cell biology. Spatial transcriptomics coupled with high throughput acquisition will be also a strong challenge. The development of deep learning and more user-friendly tools. Management and storage of hundreds of terabytes data. So this is still ongoing, and the problem that we have all of the day with the new machine and experiment. We need to improve also the machine learning teaching, and open science and sharing community is important to reach all these goals. And I think that we are all together. We agree all together with this last opinion. And what about other convergence that you have identified on this talk? And for you, which is the most critical one, for example? And what about your challenges in imaging cell biology, actually? So I will end here. Thanks for your attention. And we can open the discussion on the chat.